Welcome to Jerry's Gene Scene. In the previous videos we looked at a monohybrid cross, and we looked at how we could distinguish between homozygous and heterozygous. These are all part of Mendel's principle of segregation. Today I want to look at a dihybrid cross. When Mendel studied the monohybrid cross, he looked at one trait at a time, and in our example we looked at the white eyes versus yellow eyes of the phantasma. For the dihybrid cross, we'll look simultaneously at the color of the eyes, but we'll also look at the number of the legs that the organism has. So we have two phenotypes, one with yellow eyes and 6 legs, and one with white eyes and 4 legs. In order to illustrate our dihybrid cross, I've drawn out two models of the phantasma. One with yellow eyes and 6 legs, and the other has white eyes and 4 legs. The little dots you see are just to help me manipulate these characters and move them about. So let me just go ahead and move these guys apart so that we can create the F1 cross of these guys. So I'll move this one over to here and this one to this side. And now we'll look and see what happens when we form the gametes from these. When it comes to the yellow eyes, we know from our previous study that we're homozygous for yellow eyes, and I had also indicated to you that we would be homozygous for the 6 legs. Likewise, we can move the white eyes and we see that it was homozygous for white eyes and it was homozygous for 4 legs. What happens then is we've got only one possible set of gametes from our yellow-eyed and 6-legged phenotype, and we have only one set of gametes possible from our white eyes and 4-legged type. But let's see what happens now when we cross these two. From the yellow-eyed and 6-legged parent, we're going to contribute one set of genes that gives it yellow eyes, and we're going to contribute one set that is going to give it 6 legs. And from the white-eyed and 4-legged, we're going to give it a white-eyed. Ooh, but notice it turns out to be yellow, and we'll give it the 4-legged, and it ends up with 6 legs. So our F1 offspring is yellow-eyed and has 6 legs. It's heterozygous, but we can tell that the dominant allele for eyes is yellow, and the dominant allele for number of legs is 6. So we'll now be looking at the F2 cross, where we have heterozygous for yellow and white eyes and heterozygous for number of legs, and we're going to cross those with each other, but forming the gametes is somewhat more complex, and let's take a look at how we can do that. We know from when we did the monohybrid cross that with respect to the eyes, we could end up with one gamete that has yellow eyes and one gamete that has white eyes. And likewise with the legs, we could end up with one gamete that has 6 legs, and we could end up with one gamete that has 4 legs. But the thing is, we can also end up with some other combinations here. I'm just going to reposition these guys a little bit to further explain what I'm doing here. So these created quite by random as I was doing it. We end up with a dominant yellow eye and a recessive 4 legs as one of the possible gametes, and we end up with a recessive white eye with the dominant allele for 6 legs in this one. So we've got a dominant and a recessive, and we've got a recessive and a dominant, but we also could have potentially had a dominant and a dominant, and we could equally have created a recessive with a recessive. So now we've got our 4 possible gametes that come from the dihybrid, and each of these characteristics assorted independently. Now if we try to determine the phenotypes of the various crosses that we can get from here, you can see that we could end up with yellow and 6 legs, yellow and 6 legs, yellow and 6 legs, yellow and 6 legs, and then we can take the next one and we can see that it's yellow and 6. Oh, but here it's yellow and 4. And now it's yellow and 6 and yellow and 4. And then we check the next one out. We can see that it's yellow and 6 and that it's yellow and 6. Oh, now it's white and 6 and it's white and 6. And finally, we'll take a look at this one here, and we get yellow and 6, yellow and 4, white and 6, and white and 4. Well, that's pretty complex. So let's go and use our algorithm that we were doing with Punnett squares. So once again, we'll use our algorithm of phenotype, genotype, gametes, and offspring. And in the F1, we're crossing a yellow-eyed with 6 legs and a white-eyed with 4 legs. And the genotypes are homozygous for big Y and homozygous for big S, and homozygous for little y, and homozygous for little s. Now it's important when we're forming the gametes here that every gamete is going to have something to describe the eyes and something to describe the legs. It's not going to have 2 y's or 2 s's together. It is only going to have y, 1 y and 1 s. And in the case of our dominant, it's a big Y and a big S. And in this one, it is a small y, small s. And the only possible offsprings when we bring these two back together is that there's going to be a big Y and a little y, and there's going to be a big S, and there's going to be a little s. Make sure you keep the symbols that you're using together so that you don't get lost in the next step. So here we are, we've got 100%, as we saw, 100% that are yellow-eyed, and they have 6 legs. Once again, for the F2 cross, we'll use the phenotype, genotype, gametes, and offspring setup. And here we can see that with the possible gametes, we could have this one and this one going together. We could have this one and this one going together. We could have this one and this one going together, and this one and this one going together. So we end up with a big Y and a big S, and we end up with a big Y and a little s, and we end up with a little y and a big S, and we end up with a little y and a little s. So there's 4 possible gametes from that one, and since this one's the same, we once again end up with a big Y, big S, big Y, little s, little y, big S, and little y, little s. But now is a case where we definitely need to make a Punnett square. Let me just move this up a tiny bit so that we can get this all on. We had 4 possible gametes here, we had 4 possible gametes here, and we indicated in the previous video that you want to make your Punnett square so that it has the same number of squares as you've got gametes. So we've got 1, 2, 3, and 4. 1, 2, 3, and 4. So we could have a big Y and a big S, and we could have a big Y and a little s, and we could have a little y and a big S, and we could have a little y and a little s. You may note the pattern that I've created here is I've always gone where I've gone dominant and dominant. I've gone dominant and recessive. I've gone recessive and dominant, and finally recessive and recessive where those exist. Now we'll put these back together so we end up with the big Y and a big S and a big S and a big Y and a big Y and a big S and a little s, and a big Y and a little y and a big S and a little s. And finally on this row we've got a big Y and a little y and a big S and a little s. Now we'll fill out the rest of this Punnett square. So now we've completed our Punnett square. It's time to go through and analyze it. So once again, let's just move this up and create a bit more space for ourselves. So I'll move that up to here. Hopefully that will work here. And let me add the information that we want. We want to have a dominant and a dominant. We want to have a dominant and a recessive. We want to have a recessive and a dominant. And we want to look for a recessive and a recessive. So let's just change our pen color here for a second, and I'll use a highlighter to work this out. So this one here is a dominant and a dominant, a dominant and a dominant, a dominant and a dominant, and dominant and dominant. Likewise here we've got dominant and dominant. But here's a dominant and a recessive. Okay, another dominant and dominant, but another dominant and recessive. Here we've got a dominant and a dominant, another dominant and dominant. But here we've got one that's recessive and dominant. And here's another one that's recessive and dominant. and then we've got a dominant and a dominant, and a dominant and a recessive, and then a recessive and a dominant, and finally we end up with one here, which is recessive and recessive. Let's count these guys up and see what we end up with. So we got 1, 2, 3, 4, 5, 6, 7, 8, and 9 that are dominant dominant. We end up with one I'm having trouble seeing here. 2 and 3 that are dominant recessive. We end up with 1, 2, and 3 that are recessive dominant, and we end up with 1 that is recessive recessive. And there's our dihybrid ratio. It's 9, 3, 3, 1. So 9 dominant dominant, 3 dominant recessive, 3 recessive dominant, and 1 recessive recessive, and that's what comes from a dihybrid cross. Perhaps a little fun fact here, if we looked at the yellow eye versus the white eye, we would have a 3-to-1 ratio if we looked at the 6 legs versus the 4 legs. We'd end up with a 3-to-1 ratio. If we multiply 3 times 3, we get 9. 3 times 1 is 3. 3 times 1 is 3. And 1 times 1 is 1. So we've sort of verified that same ratio in 2 places. Remember, these ratios are predicted ratios, and we might in a future video look into the difference between the predicted and the actual ratios. In this video, we learned about dihybrid cross and the resulting 9-3-3-1 ratio. We learned that there are 4 phenotypes, and we learned about the principle of independent assortment. If you want to learn more about the monohybrid cross, check out the video that's on your right. If you want to learn more about how to distinguish between the homozygous and the heterozygous, check out the video on your left. Hit the subscribe button and be sure and like and share this video. I'm Jerry Ward. I'm at genomealberta.ca.